Good morning. I'm Joanne Geer. I'm the Executive Director of the Biopharma Research Council, and I really appreciate everyone joining us this morning. We are an association for scientists across all of the silos in biomedical research. So that's industry, academia, nonprofit, government, and supplier laboratories. We do educational programs throughout the year. Many of them are webinars like this one, although this is our most ambitious series to date. And then we have some other good things coming up, which I'll share with you very briefly. On October 28th, we have a one-day program at Research Triangle Park. And this is a really interesting kind of ecumenical, come and show each other what you're working on day. All of this is on our website, so you don't have to take copious notes. We do a series of webinars called The Mobile Patient. We're doing one on the new FDA guidance related to devices intended for the home. And in the spring, in March, we're doing a wonderful program that is our best practices. It's the sixth time we're doing it in preclinical and clinical outsourcing. And this is the beginnings of our committee, and we've got some pretty exciting things going on there. My favorite piece is that Tom McPherson is offering his wine cellar. There will be a beautiful wine tasting as part of this program, so watch for that. At any time in today's program, you can use the dialog box on the right to enter a question. Most of them will be held till the end of the program, but I will let you know two things that I always get asked is yes, this program is being recorded, and yes, the slides will be available and everything will be on our website probably by the middle of next week, and we will send out a note to let you know that they're available. A lot of people came together to make this series happen, and probably certainly the linchpin is Winston Kuo. I'm going to tell you a little bit more about him in a moment, but just to go through the 4 different sessions that we'll have. Today, we're going to hear from Jan Lotval with an overview of the exosome and what's going on in the community. Johan Skog will speak on applications on the 18th, next Thursday. The following Thursday, Sasha Vlasov will speak about commercialization. And then finally, we'll have a panel that will look at regulatory and funding issues on October 2nd. That's another Thursday at the same time. It's actually 11 o'clock a.m. And Angel is the first person on our panel. We'll probably have 1 or 2 additional folks as well. I'm going to tell you a little bit about Winston, and then he will introduce Jan, and off we'll go. Winston Patrick Kuo is the COO of IES Diagnostics, which is developing diagnostics based on a technology licensed from the FDA, focusing on systemic lupus erythematosus, and he's editor-in-chief and founder of the Journal of Circulating Biomarkers. He also sits on the scientific advisory board of Exosomes and Hansa Biomed, focused on exosome-based diagnostics and life science products, respectively. His research interest in extracellular vesicles includes the technical aspect of commercializing both from life science and clinical aspects. Winston, I'm handing it over to you.
Thank you, Joanne. Welcome, everyone. I'm delighted in collaboration with the Biopharma Research Council and NTech Publishers to provide a short course on this exciting area of extracellular vesicles. So why is this important? The field of extracellular vesicles has been abuzz for the past 5 years, and there has been significant strides both on the research front and on the commercial side as to standardized tools or methodologies to conduct those research. Just briefly, extracellular vesicles, including exosomes, microvesicles, have known to carry a variety of bio-macromolecules, including mRNA, microRNA, and other non-coding RNAs, and protein, and lipids. But within the past 5 years, I mean, it has emerged as a promising minimally invasive novel source of material for molecular diagnostics, and also involving space for therapeutics. Although extracellular vesicles can easily be identified and collected from biological fluids, I mean, much more research and proper validation are needed in order to be useful in the clinical setting. So today we're excited to have Dr. Jan Lotval, a pioneer leader with vision, who actually really doesn't need any introduction, but I'm gonna briefly introduce him to you anyways. Jan has been at Gothenburg University since 2002. He's the chairman of the Crafting Research Center, He has a clinical training in both clinical allergy and clinical pharmacology, and has a long-term experience in performing translational studies in asthma and allergy. Dr. Lawwell's research interest includes the biological function of exosomes and other extracellular vesicles, and their natural ability to transfer RNA between cells. After having published the original study documenting this process in 2007 in Nature Cell Biology. He's also the co-editor in chief of the Open Access Journal Respiratory Research, and as for the last years has been instrumental in starting two open access journals, including the Journal of Extracellular Vesicles. He's the first elected president into International Society of Extracellular Vesicles. It's definitely a rapidly growing nonprofit organization. Has as of later this year 700 delegates, and he's also a past president of the European Academy of Allergy and Clinical Immunology. So just a few housekeeping items before we begin. Dr. Lawwell's presentation will run about 30 to 35 minutes, after which we'll open up the audience for question and answer session. So you may continue to submit your questions during the presentation and also afterwards in the dashboard that should be on your screen. So thank you very much, and now I'll hand it over to our speaker, Dr. Jan Lawwell. Please enjoy.
Okay, thank you, Winston. It's a great pleasure to be part of this program, of course, giving an introductory overview. I'll just say hello to everyone, and I'll show you my poster here in the back. That is the poster from the first International Society for Extracellular Vesicle meeting, which we had here in Gothenburg back in 2012, only 2.5 years ago. We had 500 delegates. Now we're up to 700 delegates. So I'll go back to the presentation. So there we are. There we are. So I'm going to talk a little bit about extracellular vesicles in general. I will not talk so much about tissue and liquid biopsies, but actually about extracellular vesicles in general. So I come from University of Gothenburg in Sweden, and I work at primarily an asthma-focused research center, Kräfting Research Center. So as Winston mentioned, I'm a clinician, an asthma scientist that have gotten more and more interested in nanobiology, and I still think that I'm in training. So there are so many questions remaining in this field, and I'm glad we are having a lot of new people coming into this area. So this is my declaration of interest, and I have 2 patents on exosomes from 2006 and 2012 related to this presentation. So I'm not going to show so much work from my own lab, but I want to acknowledge some of the people here, Cecilia, Ganesh, and a whole bunch of other people that are actually working on exosomes in our group. Around half of these people are working on exosomes, and half of them are working in the field of asthma, translational research in asthma. I also have a good collaboration with Edith Busas and her team in Budapest, and with Yongsong Go and his team in Pohang, South Korea. So when we're talking about extracellular vesicles, we're talking about membrane vesicles that have a lipid bilayer with surface molecules often present also on the surface of the cells, and containing some of the cargo, some of the cytoplasmic molecules present inside of the cells. So the vesicles can vary in size, but exosomes that are the smaller version of the vesicles are around 40 to 150 nanometers. So previously these vesicles were called debris. I'm not allowing my lab staff to ever call anything debris, because it may very well be functional, as we're learning that these vesicles can have vast regulatory function in cell biology. So they were sort of described in 1983, and there were 2 publications that came out at the same time. There was Rose Johnston's paper together with Bintao Pan, which came out in Cell in July of 1983. But accepted and published on the 1st of August 1983 was this paper by Clifford Harding, John Heuser, and Phil Stahl. And both of these papers were discussing the transferrin receptor and its being sent out from the cells into the environment. Basically, the cells getting rid of the transferrin receptors to become erythrocytes. But if you look back further in history, there are indications that extracellular vesicles actually assisted and were important in biology. So here's a paper that was published in 1977 by Gunnar Rundqvist and colleagues showing that this enzyme was detergent inactivated, suggesting it was associated with extracellular lipids. And subsequently, they were also talking about prostasomes, actually even before the 1983. But again, even before then, this is the platelet dust that was discussed and presented already back in 1967 by Peter Wolf from the United Kingdom. So that's nanovesicles, small vesicles that are released from platelets. So there's a long history in extracellular vesicles, and it goes back maybe at least 45 years or so. But maybe we can go back even further to find even earlier description of vesicles. So when we look at vesicles using electron microscopy, you can see here they are, in this case, around 100 nanometers, and they looked a bit cup-shaped, donut-shaped even. That has been turned out to be an artifact, and when you actually look at cryo-electron microscopy, you can see that these vesicles are perfectly round, but they have different diameter. And sometimes you can interestingly even see double vesicles, and here you see both of those. And that doesn't matter if it's cell culture medium or from the circulation. So what I will discuss today is where do we have these vesicles, what do they contain, and a little bit what they can do in disease. But I will just be scratching on the surface of what they can do in disease. So if you look at the major groups of extracellular vesicles, we have in principle microvesicles that are considered to be budded off the surface of the cells through a budding mechanism right at the surface. And these can be released and contain surface receptors and can be attaching to another cell where they may have surface-to-surface interaction or may even be taken up into the other cells. Now, when we're talking about these smaller vesicles, they're called exosomes. They are considered to be produced by multivesicular endosomes, via multivesicular endosomes, where they can be filled with different types of cargo. And again, they're taken up by the other cells and go inside of another cell. So when you look at the uptake, this is our own data, uptake of exosomes. Here's using a lipid dye. You can see that the control experiment here to the left, there is very little false signal here, but actually you can see a lot of green fluorescence in recipient cells exposed to exosomes stained with lipid dye. So actually, when you look at confocal microscopy, you can see that the vesicles, each dot is not necessarily one vesicle, it's probably a group of vesicles, but they're attracted inside of the cells to one side of the nucleus usually, or sometimes a little bit more spread out. So if you start thinking about it, having a lot of different cells in the body that can produce exosomes, the smaller vesicles, they can produce microvesicles, and we also know from earlier that there are larger vesicles that are produced during programmed cell death. You have a nanocosmos that becomes quite interesting, that can influence the cell-to-cell communication quite extensively. But of course, it increases the level of complexity in cell-to-cell communication. So we should remember that the production of nanovesicles is actually a very conserved process. It's present in bacteria, for example. It's been shown in plants and parasites. So it's something that we can see everywhere in nature. It's not something that is specific for our own cells in our own body. Even there was a paper early this year in Science showing the presence of high quantities of nanovesicles, bacterial nanovesicles primarily in the ocean. And here you see the number of vesicles in relation to ocean depth of these nanovesicles. So they were also looking quite carefully in the production of this ocean bacterium and their production of extracellular vesicles. And they could take these vesicles and give it to another bacterium and see that the bacterium could use the carbon in these vesicles to sustain life. So clearly, there's a extensive amount of vesicles in our surrounding environment that may influence us and itself in different ways. This is a little bit of a provocative study that has reached some controversy, but it's showing the presence. It argues the presence of some rice microRNA in the circulation of Chinese people. And that suggests that maybe we can actually absorb RNA molecules, intact RNA molecules, via our gut into our circulation. And where in the circulation they are inside of extracellular vesicles or microvesicles. That's not necessarily plant vesicles, but actually our own vesicles, but still harboring the plant vesicles. So this has been questioned, this has been challenged, but maybe conceptually there's something there, and we have to look very carefully whether this is true at a broader sense. Anybody's drinking beer out there? I guess we are, all of us, sometimes, most of us anyway. And actually, there were two abstracts presented at the International Society for Extracellular Vesicles in Rotterdam in April this year. One from Denmark, where they showed especially that unfiltered beer contained a lot of extracellular vesicles from yeast. And another one from the my Korean collaborative group that also showed the presence of extracellular vesicles in beer. And they actually even calculated that a very large number of vesicles are ingested by humans on a yearly basis based on beer only. So when you look at the nomenclature of extracellular vesicles, there are many different names out there, and I'm not going to list all of them because that would increase the level of confusion even further. So we're trying to use this title name, extracellular vesicles, and then many people are still using the term apoptotic bodies for those that are released from programmed cell death, exosomes that are the nanovesicles, most likely to be produced by multivesicular endosomes, and microvesicles that are a little bit larger that are considered to be produced by budding from the cell surface. So if you look at the publications in the field of extracellular vesicles and just looking at these limited search terms, if you look until now, forever, almost 12,000 publications, and only since January 2012 there were more than 2,200 publications, and that means that 36% of all publications with these search terms anyway have actually been published over the last 2.5+ years, and that's pretty stunning and amazing. It's a huge growth going on in this field, and I'm very excited that many of you are still keen to work and be doing science in this field. As I mentioned before, if you look at the function of vesicles, there were a lot of different functions that were described if you look a few years back, maybe 10 years, but actually when you try to understand the cellular or molecular function of those functions of extracellular vesicles, it was very difficult to find any clear evidence. So no or very few obvious molecular explanations had been presented at that time. So now we know that extracellular vesicles can have several types of function. One is surface-to-surface interaction and principal antigen presentation, and that was first described carefully by Grassa Raposo back already in 1996. That's soon 20 years ago. Here you again see a multivesicular endosome releasing exosomes into the surroundings of this B cell. And here you can also see the proliferation of T lymphocytes upon exposure to exosomes. So they're not claiming that exosomes are more potent than B cells or dendritic cells, for example, but at least that the exosomes with antigen on their surface can actually function as extracellular vesicles. Grassa is a brilliant electron microscopist, and she actually discovered these by actually looking at pictures. That's what she tells us. But also there was over the last 10 years or so, we've learned that these vesicles can shuttle proteins, lipids, and also RNA molecules between cells. So I will discuss a little bit about the RNA, because that's, as Winston mentioned, some of the major contributions we've done over the last 10 or 20 years. So the presence of nucleotides in vesicles is not really new. It already was described in the late 90s. Here's one example of a paper showing the packaging of both DNA and RNA into apoptotic bodies during apoptosis. And we also see a lot of different RNA species present in apoptotic bodies. There was another publication coming out in 2006 which specifically focused on microvesicles, and they state very clearly that this is vesicles larger than 100 nanometers. And they are showing evidence there suggesting that mRNA and protein can be delivered from one cell to another. And in 2007, our paper came out in Nature Cell Biology that showed the presence of both mRNA and microRNA in the smaller vesicles. That's 40 to 100, 40 to 150 nanometers. I'm not going to say exactly how big they are because we don't really probably know. But we show that the exosomes actually contain and shuttle RNA between cells. So actually what we did there was we had some very complicated data on exosomes in allergen models, and we were actually we were very confused about the data, and some of the data was negative, some of the data was positive. And actually we decided to take one or two steps back and look, instead of looking at experimental animals, going back and look at cell biology and mast cells in this case. And we did proteomics and found some ribosomal proteins in the exosomes. And that made us think maybe the exosomes can be an extracellular protein factory, because they have ribosomal proteins, and maybe, but if they're going to be a factory, they also need to have the RNA. Now, asking that question, we also started thinking very quickly that they can maybe shuttle RNA from one cell to another, and it took us 2 weeks to show the presence of RNA in the exosomes, and another 2 years to actually finish the paper. I should say right now that we have so far unable to show that the exosomes are extracellular RNA factory, so a protein factory. So that's something that we have not been able to prove. Now, interesting, you can see that RNA profiles, this is using the bioanalyzer instrument, is different in that the cells have these big RNA peaks, which we do not see or see very little of in the exosomal fractions. And when we did Affymetrics and compared cellular mRNA with exosomal mRNA, we did not see a very clear correlation between the RNAs, which suggests that the RNA in the vesicles is actually specifically shuttled into the exosomes. So to prove that the RNA we struggled with that a lot. To prove that the RNA was actually functional, we took mouse exosomes from a mouse mast cell line, which we knew had mRNA, and we gave those to a human cell. So we asked whether the mouse mRNA can actually translate to proteins in the human recipient cell. So what we did is to put the mouse exosomes on the human mast cell, and we could see a lot of dots here in the 2D page of the proteomics of the recipient cells. Many of those dots were, of course, induced by proteins from the exosomes, but we could actually find a number of proteins that were present as mRNA in the exosomes, but not present as proteins in the exosomes, with it being a bioinformatics we had available at the time. So that suggested that we actually have a mouse mRNA can be taken up by a human cell and translate to mouse protein in the recipient human cell. In that paper, we also show the presence of a lot of microRNAs. We didn't do any functional studies with the microRNAs, but Michel Pectel and others did that study a few years later, published in 2010, where they, by using a reporter assay, could show that microRNAs were functional and delivered by exosomes are functional in recipient cell. Also, Johan Skog, who will be talking next week, showed the functionality of RNA by reporter assay. Now, when you start thinking about the complexity of RNA in extracellular vesicles and realize that there are hundreds, if not thousands, of different RNA species present in the mixture, the large mixture of extracellular vesicles that you harvest, how do you actually understand whether the bulk RNA is biologically functional? Well, we tested this method that was actually exposing exosomes to oxidative stress or using normal exosomes. And what we then was to put these vesicles on top of other cells and then expose those cells to oxidative stress, with the hypothesis that these exosomes would mediate a protective signal. Well, firstly, we looked at the transcripts, and this is looking at Affymetrix again. And you can see here a number of RNA species or RNA transcripts that were present in exosomes only from those exposed to oxidative stress, or those present in both type of exosomes. But we also saw a number of transcripts that were present only in so-called normal exosomes. So we had different RNA cargo in these vesicles, and that by itself, I think, was very important at the time, showing that one cell can actually change its RNA message in the exosomes they actually release. So here we see the actual data. Firstly, you can see here the blue, which is the viability in the cells exposed to oxidative stress. And here you can see quite over the 12 hours which the study was ongoing, a reduced viability with oxidative stress. But if you used exosomes that have been produced by cells exposed to oxidative stress, you could see a significant protective effect. And then we have these green values here, and for these vesicles, we decided, after a lot of discussion with different RNA researchers, to actually expose them to UV light, which is known to denature RNA. Of course, we can, at least to some degree, denature proteins, so we're not 100% sure that this is RNA, but at least this implies that the bulk RNA in the exosomes actually mediate, at least to some degree, this protective effect. Interestingly, the 2-hour protective effect is not significant, so maybe this protective effect is mediated by proteins, and this takes a bit longer and is mediated by RNA. So where can we actually find extracellular RNA? As I mentioned, I'm a primarily clinician, and we started looking into human samples, or whether this only a artifact from cell biology experiments, and we decided to look into different body fluids, and others have also looked into body fluids, and that was a big focus on that. But clearly there are RNA-containing exosomes in saliva and in breast milk and urine, and of course you can think about what happens when there's a communication via RNA-containing exosomes in kissing, or maybe even in breast milk feeding. So Cecilia, who is still working here in the lab, was first to show that human breast milk actually contains RNA-containing extracellular vesicles, and with a lot of different microRNA probably. You can start thinking about how that can influence mother-to-child communication, how that can be important maybe for the induction of immune tolerance and so on. And there's another publication from Susanne Gabrielsson that shows that human breast milk exosomes can actually induce T regulatory cells. So there is probably very exciting and important communication there between mother and child via extracellular vesicles and their RNA. One thing I want to bring up to this audience is that extracellular vesicles are present also in fetal calf serum that almost always is used in cell biology cultures. So if you look at these vesicles, you can see this is work from Ganesh Shelke in our lab. You can see that these vesicles are present in the fetal calf serum. But more importantly, we take the fetal calf serum exosomes, stain them with lipid dye, and you can see the uptake of these vesicles into recipient human cells. So, that implies that the fetal calf serum exosomes can actually be taken up. And also, when we look, and this is looking at an airway epithelial cell line, and use the dose dependency here of fetal calf serum exosomes, you can see that there's a dose-dependent increase of transmigration, I should say here, a reverse transmigration assay that was established in our lab by Yong-Song Go. So also, fetal calf serum is often used as a positive control for different experiments in cell biology. And actually, but if you use fetal calf serum, which contains exosomes, if you deplete the fetal calf serum of exosomes, in this case by an 18-hour centrifugation, you see a significant reduction in that biology, in that cell functionality that the fetal calf serum is actually mediating. So this is something to be very much aware of, and we've tried to look at how much you can reduce the RNA in the extracellular vesicles, and we have to use a 18-hour centrifugation, ultracentrifugation period to eliminate most of the fetal calf serum exosomal RNA. So some people are only using 1.5 hours, but that is not sufficient. So what can the exosomes and EVs do in medicine and in biology and in disease? And I'm not just going to touch on this a little bit in this presentation, because there's so much amazing and really fascinating data emerging right now. So as I mentioned, way over 2,000 publications only in the last 2 years, and growing very quickly. So, but if you look at the different papers and studies, they can promote disease and they can attenuate disease in different ways. So, firstly, we should look at bacterial nanovesicles, and this is some work from Yong-Song Go's lab in Pohang, where he shows the presence here of bacterial nanovesicles released from Pseudomonas aeruginosa. And so you can isolate these vesicles and separate them totally from the cell, and you can use them to look at whether they can induce pathologies in the recipient, animal in this case. And here you can see outer membrane vesicles, that are the kind of exosomes from bacteria, gram-negative bacteria. This is control and increasing doses of outer membrane vesicles given intratracheally into the mouse. And you can see a very strong inflammation, pneumonia happening in the lung without any bacteria at all. So clearly the vesicles are very potent in inducing this. And on the bottom panels you see the number of neutrophils and the number of macrophages in the bronchoalveolar lavage after the introduction of outer membrane vesicles. So these membrane vesicles can actually travel through the body very quickly. They're really small, actually smaller than normal exosomes, and they distribute efficiently throughout the body. So that's something we should consider when we think about bacterial infections, sepsis, and so on. This study showed that TLR2 and TLR4 are involved in these pathways, but not exclusively, because you cannot eliminate the inflammation totally by knocking out one of these toll-like receptors. So clearly bacteria, all kinds of bacteria, pathogenic and non-pathogenic bacteria, can release extracellular vesicles that can distribute through the body and induce an inflammatory process. So when we look at clinical sepsis, for example, we know that blood cultures of bacteria are only positive in less than 30% of patients with sepsis, even though often quite a lot of blood is actually taken. So perhaps this sepsis is induced by distribution of these nanovesicles. And actually there is evidence in sepsis that nanovesicles can be found in peripheral blood. So this is also a very exciting field. Another totally different field is thinking about tumors and cancers and malignant diseases and the microenvironment that is very complicated in a tumor. So here you can see that the tumor is containing, of course, cancer cells, but also many other types of cells, fibroblasts, immune cells, mast cells, sometimes eosinophils, endothelial cells, and so on. So if you think about all of these cells actually being able to send out some type of nanovesicles and influencing other cells in the environment, you realize that this mesh of communication between cells in the tumor microenvironment is very complicated. So there are a number of studies that actually look into the consider the tumor microenvironment. And for example, exosomes secreted under hypoxia can enhance invasiveness and stemness of a prostate cancer cells. So that suggests that the cells in the center of the tumor that live under more hypoxic circumstances actually release different types of vesicles that have different types of function. And here you see invasiveness, the difference here induced by the different types of extracellular vesicles, or exosomes in this case. Also, this study from Lorraine O'Driscoll from Dublin shows that exosomes from a not so proliferative versus a proliferative tumor can actually, the proliferative tumor exosomes can induce enhanced proliferation in the recipient cell. So that suggests that actually proliferation can be enhanced if it's sent from one cell that is prone to proliferate to another cell. One of the most interesting and mind-boggling studies recently published in this field is this study that came out in Stem Cells in March this year, which suggests that adipose stem cells can be reprogrammed by prostate cancer cell-associated exosomes to actually produce tumors. I think this is, of course, really interesting because that suggests that exosomes can actually shuttle tumor-like molecules or tumor-inducing molecules from one cell to another cell. And maybe cancer is then polyclonal, and partly the polyclonal process is mediated in part by extracellular vesicles. We are also looking into mesenchymal to epithelial mesenchymal transition in our own lab. We can see that mast cell exosomes, in this case looking at airway epithelial cell line, can induce a mesenchymal transition. And there were another group in Canada that showed that, published in Cell late 2012, if I remember correctly, showing that Wnt signaling may be involved, but also other pathways may be involved in this functionality. So one of the big problems or big issues that we in the extracellular vesicles community and field have is that there are many subsets, and we often isolate different types of vesicles. So firstly, I mentioned that we have exosomes, we have apoptotic bodies, and we have microvesicles, and those are the big subgroups, and maybe there are subgroups within those subgroups as well. So last year we published a paper together with Edith and Irma in Italy. The first author is Cellular, where we looked at the RNA profiles. We didn't sequence here, we just looked at the RNA profiles using the bioanalyzer of different types of vesicles. And if you look firstly at the exosomes, these are pellets in the plastic that are sectioned. You can see here nanovesicles, and here this is 3 different cell lines: the human mast cell line, a human erythroleukemia cell line, or a mouse microglia cell. So fundamentally different vesicle, different cells. But all of these cells release some sort of smaller RNAs without the ribosomal RNA peaks. Microvesicles look a little bit different in the electron microscopy, but in this case we could not detect any RNA. Here we can see some ribosomal RNA peak and little small RNA, and here we can see no or very little RNA. Whereas you look at apoptotic bodies, again looking totally different in the electron microscope, we see quite prominent rRNA peaks. So that suggests that these types of vesicles have fundamentally different RNA cargo in them. We pursue this further by Taral Lunavat in our lab to look at melanoma exosomes, and this is looking at melanoma cell line. But here you see the RNA profiles from cells, from apoptotic bodies, and from microvesicles, and from the nanovesicles, exosomes. And you can see that the RNA profile is very different in the exosomes compared to the other types of vesicles of the cells. And actually we see a lot of microRNAs in the exosomes, and this is short RNA sequencing in collaboration with Andy Hill's lab in Melbourne. We see a lot of small RNAs here. And when we look at microRNA specifically, we see a number that are unique for the exosomes. And here you see a number that are commonly seen: 451A, 155 is there as well. Whereas the microvesicles and the apoptotic bodies have no or very little, few unique macroRNAs. So when you look at the cluster analysis, it seems that from the RNA perspective, that the exosomes, the microvesicles, and the apoptotic bodies actually are different, although we cannot exclude the possibility of overlap between these two types of vesicles. Cecilia Lesser in our lab are now dissecting the possibility of 2 types of exosomes. So when we looked at our first RNA data, we saw no correlation between the cellular RNA and the exosomal RNA. But in some studies, this is a study from PLOS ONE a few years back now, 2 years back, in some cases they see a very high correlation between the mRNA in the exosomes and here and in the cells. So this made us a little bit confused. And why is one study showing no correlation and another study showing good correlation? So to us that suggests that maybe there are 2 types of vesicles, and actually we are now able to separate 2 types of exosome-like, I should say, extracellular vesicles. We call them E1 and E2 here for simplicity, where one has a good correlation of mRNA, and one still has a significant correlation, but it's not as clear as in this one. So that suggests that there are subsets of nano-sized vesicles that have different RNA cargo. Also, the vesicles are protected from a proteinase K and subsequent RNase. This is work by Ganesh in our lab. Whereas in some of the other vesicles, maybe the RNA is not totally protected by the cell membrane, because proteinase K followed by RNase will reduce the RNA. So that suggests that some of the RNA is not protected by the lipid bilayer. So when Cecilia went further, she saw that one of the vesicles were smaller, significantly smaller, and the other one was significantly larger. This is counting individual exosomes. You can see a significant difference here. And also when we looked at proteomics, we see some overlap, but there are unique proteins in the E2 versus the E1 vesicles. And I should say now they were separating these through creative filtration and centrifugation assay. So maybe this nanocosmos that I mentioned is much more complicated than we have considered over the last few years, and maybe we have subsets of vesicles. Maybe these overlap. Maybe within one subset we have several types of vesicles, and it's becoming very, very exciting and interesting to try to understand this complexity. And also, when we take two types of vesicles, this is not the E1 and the E2, but we can actually see a different response in recipient cells. This is again looking at transmigration in recipient cells. So one vesicle from a cell and a different vesicle from the same cell may have different functionality. So one of the exciting fields, this is just the last few slides, is that exosomes putatively can be used as therapy. And this is one of the stronger papers from Tokyo National Cancer Center, which produced an exosome with a peptide that attracts to the EG receptor. And when they load them with LET7, they can see an inhibition of tumor growth, suggesting that one, you can target exosomes at least partly to tissues, to tumors, but also you can deliver functional RNA to those tumors. Excuse me. So in summary then, I think this field is extremely exciting. The exosomes and other extracellular vesicles contain RNA, or carry RNA. This RNA can be shuttled between cells. Excuse me. They can be mediate biological messenger and alter the phenotype of the recipient cells. I'm not going to talk so much about biomarkers, but maybe they can be used as therapeutic vectors, and you can putatively use these for treating both cancer and inflammatory diseases. So last but not least, I would like to second what Winston said, and that is welcoming you all to the International Society for Extracellular Vesicles meeting in Washington, D.C. in April next year, which is at least partly is being organized in collaboration with the NIH, although it is an ICEV meeting. And also we have a one-day course together with this meeting happening in San Diego in California in only a month and a half away. So again, thank you very much for your attention. This was very exciting. Now throw questions at us and start the discussion. Thank you.
Thank you, Jan, for such an outstanding and informative presentation. So for those that are remaining on the line, please continue to submit your questions. Even if Dr. Lotwell does not answer them today, we hope to provide a link with the webinar as well as the written responses to the questions. So Joanne, do you want to take the lead in terms of the questions?
Thank you, Winston. Yes, we've got some great questions here. I really appreciate this. Um, we will begin with, I'm going to take them kind of in not any particular order because they've been entered through the whole process here. We know that both miRNAs and mRNAs are present in EVs. We also know that miRNAs target and degrade mRNAs. Is this also happening in the EVs? Is there any evidence about this?
So that's of course a very interesting question. Firstly, the first big question is what happens within a single vesicle. How many nucleotides can actually a single vesicle contain? And I think Sasha Vlasov will discuss this in more detail in 2 weeks because he did some calculations a few years ago saying that only 1,000 or if it was a few thousand nucleotides can exist at the same time in the single vesicles. We have no idea about the RNA cargo in individual vesicles. So when we're looking at all this data, we're looking at a mixture of many vesicles, and that's a which I say makes it more complicated to answer a question like that. But I think it's a very valid and interesting question. Do the mRNAs and the microRNAs actually travel together in single vesicles? Next generation experiments will provide that answer.
Great. We have another question. What is the significance of nuclear polarity of exosomal uptake?
So actually, we don't know that yet, and it's an interesting observation, and we and others are and others are actually pursuing this work. We are kind of interested in the different types of uptake pathways of different types of extracellular vesicles, and we're pursuing some of that work. So I remain to see a paper which actually explains that importance. But the rapid travel of exosomes in towards the nucleus is something that has been mentioned repeatedly in different meetings.
Okay, and from the same person, we have another question, so we'll continue
You
on.
you can introduce a person. That's fine.
This is Alan Ezrin.
Oh, I know him. Hi, Alan.
Is it plausible that RNA susceptibility to protease K in E1 and E2 MCVs reflect lipoprotein-trapped exRNA that are aggregated but not internal to exosomes?
So this is cell supernascent, and I don't think we in the proteomics see any lipoprotein or very much lipoproteins, certainly not complete lipoprotein proteomes. So, but clearly there is some sort of, I'm not excluding it, but there's some sort of extracellular, extravesicular protein complex that is probably protecting the RNA, yes.
And Alan says hi.
Hi.
And I hope I'm saying this right. Juliana Silveira from Brazil. Dr. Lotfeld, would you talk a little bit about the controversy of miRNAs being outside and not inside of EVs?
So yes, so we are very interested in that, and we are really pursuing that work ourselves right now, and we haven't necessarily published all the work, but in our hands, as I showed you, the most of the RNA in the exosomal isolate seems to be protected from a proteinase K followed by an RNase, and that argues that because the proteinase K at that concentration will not destroy the lipid bilayer. So that suggests that the RNA that is inside of the vesicles is protected, and that's where a lot of the microRNAs are present. So again, I think it connects a little bit to the first question. So how do we actually show the presence of the RNAs inside of the vesicles versus outside? Somebody needs to dig into that and really try to eliminate everything that is outside of the vesicles and then inside of the vesicles. But I assure you, we are actually right now pursuing those experiments. So soon we will have some answers to you. Maybe at the ICEV, for sure, at the ICEV in Washington D.C. we will have at least an abstract on that topic.
Great. Tom Fair asks, how quickly or slowly are exosomes cleared from the body? What is the rate limiting step?
So this is of course an extremely important question, and so there are two issues here. Firstly, how stable are the vesicles? How stable is the RNA cargo in the vesicles, and how long can they stay around in a liquid? Maria Eld in our lab did some experiment by mistake. To be quite honest, she actually took exosomes and put them in an Eppendorf tube and put them in the refrigerator and forgot them there for 3 years. 3 years. And then she thought, oh, maybe there is RNA still in these vesicles. And yes, there was intact RNA in those vesicles after 3 years. So that shows that the vesicles are exceedingly stable in an extracellular environment. Now, when you put them into the body, the half-life seems to be very short. So when they go into the circulation, they are probably taken up by both tissues, maybe cells in the circulation, maybe endothelium. So they will probably clear from the circulation very, very quickly. Now, when an exosome is produced in a liver cell, and if you ask how fast that is sort of leaving, how fast that exosome is disappearing from an extracellular space, that's extremely difficult to say. But probably the release and the uptake is happening quite extensively at a local level. So you have very short distances between cells. You have a cascade of vesicles that are released from one cell, and very rapid uptake of the cell that is just adjacent to that cell, to that producing cell. So those are the dynamics you have to think about when you're actually looking at this. But of course, if you look at therapeutic effects, you have to look very carefully into the pharmacokinetics of the vesicles and their targeting capacity.
Thank you. We will take one, we do have one or two more questions, but I'm going to launch a short poll that I was supposed to do at the beginning.
Yeah.
I'm launching it right now, and you'll see we're just asking, are you currently working with exosomes? And there's a couple of different options there. So if you'd weigh in, we'd appreciate that. Let's see. Bear with me a moment. More questions coming in. This is great. Let's see. Robin Chowdhury is saying, given their small size and difficulty visualizing individual exosomes, what is the best method to track their distribution? For example, following injection.
So I think Cassandra Brakefield had a paper I only recently that suggest you can actually track them in vivo, but I have to remind my memory to actually look into this in detail. I think there are many different possibilities, obviously, that you can look at the functionality of them. You could look at if you could load the vesicles with some type of RNA molecule, and you have a report in every cell in the body. You can inject that exosomes and see where you see the knockdown in the recipient cell. And I think that's actually the most interesting and functional distribution assay you can think of. Just looking at imaging is only imaging. It's not showing anything on functionality.
Okay, I'm just one or two more. We have a question from Arpang Koi Wang. So, and this is related to the cells that were left in the fridge, which I think is a great story.
Exosomes, yeah.
Sorry. So would you suggest that samples, for example, cell culture supernatant, to be used for exosome isolation can be kept at 4 degrees Celsius for a long time?
I'm not saying that. I'm not saying that at all. And I'm just giving you an example of how some vesicles, in this case, I think it was from a mouse mast cell line, I'm not 100% sure, can actually retain their RNA profiles for a long time. I'm not saying that their functionality is retained, and there is debate going on within the community whether you should freeze your vesicles before, if you want to do functionality later, or if you should keep them on ice, or if you could even keep them in room temperature. So I think that that depends on you. You have to do the experiment yourself. I'm not saying that's necessarily possible, but we can freeze them without cryopreserving them. That's clear, and you can retain functionality without cryopreservation molecules, chemicals.
Well, thank you very much, Jan and Winston, and everyone who has participated today. I'm just going to read the results of our poll. We have 30% of folks who've just getting started. 44% have been working with exosomes for a while. 7% are saying not right now, but they might, and 19% are just interested. So it's a
Great.
great distribution.
Perfect.
Do you have any final comments, gentlemen, that you'd like to make?
Winston, go ahead.
Oh no, I just want to say thank you for the participants, and thanks a lot, Jan, for a very informative and enlightening presentation. And I just want to remind everyone, this is the first session of 4, so please don't forget to register for upcoming sessions. And thank you very much for participating.
And thank you very much for inviting me, and thank you to the whole audience for staying on the whole period. Thank you.
Thank you. Be well, everyone. We'll see you in a week.
Thank you.
Bye.
Bye.
Bye-bye.